All right. Thank you, everyone, for joining us. This is our August FISC PAC, or Florida Invasive Species Council Partnership Assistance and Communications Committee monthly SISMA call. And we're here to kind of share out information and help Florida's cooperative invasive species management areas, or SISMAs, as we work across the state to improve invasive species management. And so for today's presentation, we have a special topic. We're looking at student research presentations, and we're working with some of the affiliates from UF's New Invasion Science Research Institute. And we've got a really broad suite of topics today that I'm pretty excited about. And we're covering some areas that are a little bit new or different for our group. So we've got topics looking at agricultural invasives. We've got looking at our native species that have gone on to become invasive elsewhere, as well as looking at different invasive species here in Florida. And so I think this is great for us. Expanding our knowledge a little bit helps us have more vocabulary and more reasons as we're talking to our public citizens about why invasive species management matters and how invasive species can affect our lives in many different ways. And so with that, I'm going to go ahead and hand it over to our first speaker, Jacob.
Thank you very much, Deb. So can you all hear me okay?
Yes, sir.
Okay, I'll share my screen. So can you guys see the full screen now?
Not yet. It may just be taking a second to load. Still not coming through. You want to try again?
Yeah.
All right.
Okay, is it working now?
Yes.
Is it in full screen? Okay. So hello, everyone. Today I'll be talking about an invasive fungus that specifically attack bananas. So this is a really interesting story, and hopefully we're going to have an interesting insight regarding why this fungus can threaten the food security of a whole country and also the whole continent. So the outline of the presentation is first, we are going to have a quick overview regarding Fusarium wilt of banana. So this is the name of the disease, and FOCTR4, which is the fungus that causes this disease. And finally, why it threatens food security in Colombia, which is our case study, but it is not restricted to Colombia. The second section is how we can use expert knowledge elicitation as a valuable tool for gathering hard to obtain data. So some of the factors that are associated to the dispersal of this fungus are very difficult to obtain. So we use this methodology. And finally, with this information, with the network of planting material trade and the network of stakeholder movement, how the dispersal of this pathogen or future invasions of this pathogen can be simulated and how management practices can be implemented regarding that future scenario. So first of all, I want to give a brief introduction of this disease. And basically, the Fusarium wilt of banana was one of the most important plant epidemics in the 20th century. So this is a well-known history, maybe for plant pathologies, and some of you might know about this story. So this disease was first detected in the 1890 in Panama, and 70 years after that, it was widely spread in Central America, South America, and the Caribbean. So one of the reasons why this disease was widely spread was because before 1960s, the cultivar Gros Michel was widely used, and this cultivar was susceptible to this fungus. And some of the data says that in 20 years in Honduras, 30,000 hectares were lost, and in Costa Rica, 6,000 hectares were destroyed because of this pathogen. But why is this pathogen so important and why the spread of this disease was so quick? So first, the pathogen is called Fusarium oxysporum, forma specialis cubense, race 1, and this is a soil-borne pathogen. So one of the important features of this pathogen is that it can last for over 30 years in the soil. So once you have this pathogen, this fungus in your field, it's going to be extremely difficult to get rid of that. Also, because it's a soil-borne pathogen, every aspect regarding the movement of these soil particles could be influencing the spread of this disease. So that means that, for example, wild animals, domestic animals, like vehicles, workers, tools that the workers are using, and water, all those are factors for spreading this disease. And also, importantly, planting material. So these bananas are vegetatively propagated. So if you grab one sucker from this infested area and you plant that sucker in a different area, you will be spreading the disease. So that was history because the industry, the banana industry, was able to overcome this situation with a very fancy way, which was changing the Gros Michel, the susceptible cultivar, for Cavendish, which is a resistant cultivar. So basically, the solution was that fancy that people were not thinking about this problem anymore. But it was costly because the fruits are different and also the methods for packing these bananas were different. But the honeymoon didn't last too much because in 1990s, a new race of this pathogen was identified in Asia and in Oceania, and then this new race was able to infect Cavendish banana. So this golden cultivar that was used in this new era of production of bananas, it was now susceptible to this new race, the TR4. So as you can see, the TR4 race of this fungus is the most pathogenic race, and it has a wide range of hosts. But, you know, in the Americas, this was not a problem because, as I said, the pathogen was restricted to Asia and Oceania. But basically, in 2019, this invasive fungus was detected in Colombia. Then in 2022, it was detected in Peru. And then in 2023, it was detected in Venezuela. So somehow this pathogen was able to come overseas and establish compatible infections. And now it is spreading around South America. And something interesting is that the isolates that are being detected in these different countries are different. So it is suggesting a different introduction of this pathogen. So one of the first questions that people, specifically in Colombia, were having was regarding plantains. So banana is a really important industry for the international market, but plantains are a staple crop, which is crucial for the food security of the country. And these plantains that are planted in Colombia are resistant to the race 1. So the first question was, okay, so what is the behavior of these locally grown plantains to the race 4? And basically what we can see here is that all of them are susceptible to this new race of Fusarium. So basically, the whole industry of bananas for the international market in Colombia is being threatened. And also the food security associated to plantain is being threatened because of this pathogen. And I can say that, as you can see, the production of bananas is limited to 3 or 4 departments in Colombia. Departments are like states. But plantain is widely produced in all the departments in Colombia, and the production could be 6 or 7 times greater than the production of bananas. So basically, that's why we say this is a major threat. So as I said, because this is a soil-borne pathogen, there are some factors that are associated to the spread of this fungus, and some of them are the planting material and the movement of stakeholders associated to the soil particles that they can move through their shoes, and the equipment, and the tools and cultural practices they use. But how can we gather this information? When we are talking about trade of planting material in a developing country, and specifically in banana and plantain systems, the informality is absolutely huge. So there is no databases regarding how this planting material is being traded, even though it is the most planted fruit crop in the country. So for that, we use expert knowledge elicitation, which is a tool in which we go to those places and we ask experts regarding that information, and we try to build some initial parameters to identify what could be the possible dispersal of this pathogen. So basically, the two objectives of the EKs, the expert knowledge elicitation, were to map the stakeholder networks and planting material trade networks, and then develop simulations and what could be the impact of different measurements in order to reduce the future invasion of this fungus. So these are some of the examples that we have from the EKs. So we were talking to the experts, and we were building the network of planting material in plenary sessions, individual surveys, and focus group discussions. So going to the results, we are going to see the trade network of planting material. And for that, we need to first understand what is a network. So in a network, we have nodes, which represent places. In this case, are departments, which are like states. And the links, which are these arrows here, are indicating any type of link. So in this case, if we are talking about trade network of planting material, node 2 is trading planting material to node 1. And the node 3, which is a place, it is trading to node 1, but the trading is not going in the other way. So what we see here is that we have an intricated network of trade of planting material, and these sections here, these stars here, are indicating the places where the pathogen is present. And we see that we have some clustering, some aggregation of the network by geographical regions, and these regions have different behaviors. So we are identifying also the places that serve as bridges between these clusters, and we have some interesting features in these different clusters. So for example, here we are having a certain a connectivity with Venezuela, and this cluster over here is characterized for having intercropping systems. So, but what are intercropping systems? So they are these systems in which we are mixing coffee and plantain, or plantain and another species. So from the epidemiological perspective, doing management practices in these fields is more difficult. Also, looking at the movement of a stakeholder in the banana and plantain systems. So again, we see a dense network regarding this specific variable. And again, we see that these places are those in which the fungus is right now. And we are looking at the map, at the clustering here, and we see that this group here is being connected to this group, which is having a geographic, which is not sharing geographic boundaries in this case. So what we are concluding here is that basically through the movement of people in the systems, we can connect regions which are geographically disconnected. And we have also some regions which are having connectivity with the neighboring countries, such as Ecuador, and some regions which are having high international connectivity, specifically because they sell those bananas in the international market. So the next and most important question that you can say is that, so what is the future? What is the future invasion or the possible dispersal of this pathogen if we have these networks? And in this case, we can see that if we simulate, if we decrease the connectivity of the networks that we saw before, we can see also a decrease in the dispersal of this pathogen. But this a decrease in the dispersal, it's only efficient if we are decreasing the connectivity of both networks, the network of trade of planting material and the network of stakeholder movement. So that means that we have two dispersal parameters, the planting material and the movement of stakeholders. And basically what we say from the practical perspective is that you have to tackle both in order to reduce efficiently the dispersal of this pathogen. So basically that's the main take-home message that this research is trying to say. So if we intend to decrease the invasion of this pathogen from these two locations where it's present, we need to perform management practices in order to reduce the connectivity of the planting material trade network and the movement of stakeholders in Colombia. So basically, that's what I have for you all. And if you have questions, feel free to ask.
Thanks, Jacob. But seeing that and thinking about the different networks, it's a new way to kind of, I think, approach invasive species management. It makes me think of here in Florida, two of our big species are old world climbing fern and Kogan grass, and those get moved around on equipment and by people, and could be in things like pine straw, and also it'd be interesting to see something like that applied to some of our management issues here in the Southeast. As far as like getting the stakeholders involved, did you find that people were interested and willing, or was there some reticence in trying to get involved in kind of figuring out how this moved?
So basically, because we are talking to experts, most of the time they are very willing to give you that information because they know the importance of knowing this in a greater picture, in a big picture. So we didn't have that like challenge because we selected very well the people that we were taking data from. And I think that probably that could be a suggestion if you want to implement something like that, trying to identify very carefully the experts.
Oh, very cool research. Any questions from the group? All right, if you have questions, feel free to throw them in the chat. But in interest of time, we'll go ahead and move on to our next presentation. Oriol. You're still muted.
I try now?
Yep, I can hear you and I can see the main screen.
I tried. What do you see now?
I see it's not in slideshow view, but I just see your slide.
Okay, I continue like this because I cannot make it work.
This will work.
Okay, so this project is between here the University of Frankfurt in Germany and the University of Florida, and we are trying to discern if invasive populations of fish have a greater tolerance compared to the native ones in the US. And for this, we used the mosquitofish, which is a widely spread species all around the world, and exposed to nitrite, which is a toxic widely present in eutrophic areas. So human activity are altering the nitrogen cycle by putting more availability of nitrogen by, for example, manure spreading, the water treatment plants, runoff from agricultural areas, and the use of nitrogen fixing crops. And this is a worldwide problem. Here in Florida, I remember the Lake Apopka, Okeechobee is suffering from these problems. And this greater availability of nutrients causes the eutrophication, which by decreasing the complexity of the trophic web and the biodiversity is a hotspot for the developing of generalistic species such as mosquitoes, so that they are able to hold in low oxygen waters, very polluted. And these mosquito populations, as you know, is a malaria vector. Nowadays, only especially in tropical areas, such as South America and Central America and Africa. But in the 19th century, it was a big problem in the US. For example, here you can see on the left top that it was widespread from the Midwest to the eastern side. But by the consequent use of DDT and other biological control, it was controlled in the mid-50s of the 20th century. One of the measures or the anti-malarial works, it was the spread of the predators, and it turned out that this fish was selected as after some experiments in the early 20th century, it was concluded that this fish had a lot of anti-mosquito activity. So it was spread. So here in yellow, you can see the native area for Gambusia affinis on the west side, and Gambusia holbrooki here on the eastern side of the US. And here you can see that was introduced to the western states in the US, but also Hawaii, American Samoa, Guam, and now is present in all continents except for Antarctica, in all the places where where malaria was a prominent problem. So what the US authorities, after the petition from the European countries, they shipped a container of mosquito fish to Europe. But during this trip, most of them died. So at the end, only 6 individuals were introduced in Spain. So there was a huge bottlenecks or reduced genetic variability. But these 6 fish started to reproduce and populate different ponds in Spain. And from Spain, it was shipped to Italy, from Italy to France, and from Italy to the former Yugoslavia, and all around the Mediterranean basin. Here I cannot show because it's supposed to rotate this figure, but you can see in blue that all around the Mediterranean basin and also in Africa and Australia, it is widespread. But the question is why this is so successful, this species, even though the genetic reduction after the introduction event. So one of the hypotheses is that because this fish was introduced mainly in eutrophic areas where mosquitoes were present, it has developed or evolved a tolerance to nitrite or ammonia and nitrite. So all those nitrogen pollutants that appear in eutrophic areas. And in this experiment, we selected nitrite as the test compound because it is much more toxic than nitrite and is more stable in the water compared to ammonia. So ammonia is rapidly oxidized to nitrite, but also nitrite is rapidly oxidized to nitrate, which is the less toxic. So the general objective was to assess the toxicity of nitrite on the eastern mosquito fish on individuals from 9 different populations, from Florida, North Carolina, where it is native, and Extremadura in Spain, where it is invasive, at varying background nitrogen pollution. So the methodology for this experiment was to get wild-caught mosquito fish from 3 different sites, first in Florida, and we selected the sites based on the nitrogen pollution. Here you can see on the picture, this is Ruth Springs near Brentford, but also we fished on Poe Springs, which is supposed to be relatively cleaner. or less polluted than the other sites, although nowadays it's difficult to find a really clean, unpolluted site. And we acclimated them in the lab at clean, fresh water with daily feeding. We repeated the fishing in North Carolina. So the fish sent to Europe, they came from North Carolina, exactly the Edenton hatchery. So we got fish from the Saar population in Edenton, then Newell Pond and Albemarle Canal, that is supposed to be more polluted. And finally, here is the Spanish sites. That this spring that you see in the picture is the place where they did introduce these 6 fish, and where they reproduced, and then there was spread on all the other sides. So after acclimation from each site, some fish were exposed to nitrite, and other to the negative control. And after 10 days of exposure, we assessed different endpoints to see how the pollution was affecting the health of the fish, and if there was a different response based on the origin of the fish. So we evaluated the respiratory rates, blood parameters, gene expression, and embryo development. But because of time restriction today, I will only show the ones highlighted in red. So here in the respiratory rates, the oxygen depletion after some time of the fish in a tube was measured to get the rate of oxygen consumption as a proxy for metabolic rate. And here you can see that the control fish is in blue, and the exposed fish in the lab is in red. Here you can see that the Florida fish don't change the metabolic rate, while the North Carolina decrease the metabolic rate when they are exposed to nitrite. So there is an effect in the North Carolina fish that is not seen in Florida. When we tested the Extremadura invasive fish, the pattern that you see in North Carolina is reversed. So they increase the respiratory rate. Remember that the Extremadura fish is coming from the North Carolina fish. So genetically, the Extremadura fish are closer to the North Carolina compared to Florida. There is something unusual in the Florida fish, that in the springs, the oxygen concentration in the springs is very low, because it comes from groundwater. So this fish in Florida might be adapted to low concentrations of oxygen, and because nitrite oxidizes the hemoglobin of the fish, there might be an interaction between oxygen available in the environment and the effect of nitrate. Here you can see, the North Carolina decreases and Extremadura increases. So because of this effect of nitrite on the hemoglobin, which, remember that the hemoglobin is the responsible of transporting the oxygen to the different tissues of the fish, we also looked at the hematocrit to see if there were differences. Again, here you can see that nitrite increases the red blood cell count. So it increases the hematocrit of the fish. So there is a general pattern in all fish, regardless of the region of origin, that is conserved. But in Extremadura, we find higher baseline red blood cell count. So there is a higher hematocrit, regardless of if they are exposed or not. So there is a difference in the physiology regarding the region of origin. So when evaluating possible effects of an invasive species on the environment, the origin of where they come from and the possible adaptive mechanisms that appear during the introduction, during the invasive process, might appear and might be taken into account. It is remarkable that the fish were introduced 100 years ago. So during these 100 years, there must be some mutations, some adaptive phenomenon to the local conditions. So this has to be taken into account also. So we also looked at the embryo wet weight, which is decreased after nitrite exposure in all fish. So there is a general effect of nitrite in different physiological pathways. And here you can see also that the Extremadura fish and the North Carolina, which are the ones that are closer on the genotype, they have smaller embryos compared to the Florida ones. And this is important in terms of ecosystem effects or fitness effects, because the size of the fish can have important consequences. So more susceptibility to predation, or usually fish, if they have lower, so smaller embryos, they have higher number of embryos. So the patterns of number of fish that can colonize new environments in the invasive areas is higher, or can be higher. And yeah, this will be compared, because to talk about genetic differences, we are also looking at the genetic expression of these exposed fish. And these results are incomplete until we have these genetic data. And thank you for your attention. If you have any questions regarding this.
Thanks, Oriol. So just want to make sure I got it right. 6 individuals are all that survived, but they were enough to, when released into the spring there, those 6 individuals were enough to start a self-sustaining population, right?
Yes, these 6 individuals, after they were released, after the winter, they were self-sustaining and were colonizing the adjacent areas from this spring.
That's I would not have expected one introduction of 6 individuals to be enough to build a population. That's kind of scary thinking about what might be making it to our shores.
Yeah.
But thank you very much. In the interest of time, we're going to move on to Sarah.
Yeah.
And if anyone has questions, feel free to throw them in the chat, and we can answer them via the chat, or at the end if we do have a little extra time left over. Sarah, you still with us?
Yes.
Awesome.
I'm not able to share it yet.
Let me. All right, you should try now. I see the slides.
Okay. Okay, so hi. I'm Sarah Salgado McDonald, and I'm working with biological control in the Mintier lab in Fort Pierce. So we're gonna move a little bit from fish to a plant. So this is earleaf acacia, and is what we call an emerging threat to Florida's ecosystem. This plant belongs to the acacia family. Acacia is well known for its invasive species. They have around 24 plants that are invasive around the world. And these acacias are in the legume or Fabaceae family. So it's a pretty important family of plant. So this is a fast-growing evergreen tree, and it's native to Australia, Indonesia, and Papua New Guinea. So this plant, earleaf acacia from the other side of the world, came to Florida in the early 1930s, and it was introduced as an ornamental plant, just like all the plants did back in the day. Since its introduction, it has been found in disturbed and wildland bordering areas. So these plants have some traits that helps to the invasiveness, such as allelopathy. This is a fast-growing tree. Just as an example, we have a field plot that we planted in 2019, and within 2 years, these plants were able to reach up to 4 meters. So they grow very, very quick. They also have long leaf flowers. As we can see, these bright yellow, beautiful flowers, and that helps to have more pollination, to be exposed to pollinators. And they also have early reproductive maturity. So these trees can start producing seeds within the first year. This is an example of how the seed pods looks like. The picture on the left, those are the pods when they are young. So they are green. It's a bit hard to spot them when we're looking at the trees on the field. But once they become mature, it's easier. So they turn this brown, leathery texture, and they open, and the seeds are going to be hanging around. These ones are the seeds. And that's going to help them to be dispersed by birds. So in Florida, we know the current distribution. So they are in the central and south Florida. So in this part of the map, we don't have records yet, but we don't know if it's because they haven't seen it, or if the plant it hasn't made the way there yet. So if you're driving in Florida, you're often gonna see these trees along the road. This is how they look like. They're big trees. And if you are around here and you have some time, please report this tree in iNaturalist. So we created a project that is called Earlification Florida. So we're trying to see the distribution of this tree, where it is at, and that will help us later for some management. If there are areas that the tree is not there yet, we can try to prioritize these areas for integrated pest management. So if you see this tree, please report it in iNaturalist. So we know that the tree is here, and we wanted to see what areas they can expand to. So we created this model is based on climate data. And the areas that we see in red are the areas that are highly suitable for earlification in Florida. And the blue color, it's the areas that are not suitable. And then we use the records that we know just to have some something to relate to show that our models are good besides the numbers. So this is what we expect in the field. And as we can see, this part here, we don't have records, and this belongs to the Everglades. So again, the Everglades is a bit more trickier to access because it's around 500,000 hectares of subtropical fresh water. It's a wetland or a river of grass. So the Everglades had been invaded, has had many problems with invasion. So we're trying to see if the tree is there, or if we can still prioritize these areas. So what's happening with the control for earlification? Currently, we are only working with mechanical and chemical control. Mechanical, just like if you see the trees, you're pulling them down, hand pulling the seedlings or cutting mature trees, or just applying chemicals. But looks like this is not enough yet because the tree continues to expand. So right now we're working on a classical biological control program in which we have 2 insects that are being under studies. The first one is a Calomela intermerata, is a leaf-eating beetle, and the second one is a bat-gulling wasp. So for this biological control program, it's not something easy. So first we need to start selecting our target, in this case, earlification. We start doing some native field work, looking for co-evolved natural enemies that are feeding on the plant and causing significant damage in their native range. And then we select them. We bring some into quarantine. In this case, we're working with 2 quarantines. One is here in Fort Pierce with University of Florida, and the second one is in Fort Lauderdale with the USDA. In quarantine, we do a bunch of studies, one being a host range. That is one very important because we select a list of plants based on the target plant that we have. We use plants that are very close related. We also use plants that are in the same genus, in the same family, in the same tribe, plants that have a similar biochemicals, chemical compounds, plants that are native, are endangered, and plants that have economic impact in here. So after that, and if they are a host specific, meaning that they were only feed on this target plant, we're gonna continue with some paperwork that we call regulation. And once that is approved, we continue to field releases. And we also work with some technology transfer or collaborative efforts to integrate our biological control agents into integrated pest management programs. So all of the, in this case, we are currently with earlification in the step 3. We have 2 insects in quarantine. And all of this pipeline, like we call it, takes about 8 to 10 years to complete. So we have a lot of work that we have done and a lot more that we have to do. So all of this started with this research. This paper by Mintier and collaborators, they started with field studies in the native range in Australia. And they brought these 2 insects into quarantine that I mentioned before. And now I'm going to talk a little bit about each one of them. So the first one is Calomela intermerata. This belongs to the Coleoptera or Chrysomelidae family. It is from Australia, just like the plant. We have 2 biotypes from 2 different provinces in Australia. They feed on leaves of earlification, and they were imported into quarantine in 2018. Since then, we have done some studies, one being a host range. In this test list, we have around more than 100 species, 115 plant species. 80% of these species are done, being that we tested for larvae and our adults on feeding on these other plants. It's almost done. The tag petition is going to be filed in June 2025, so it's very close. So far, they do not reproduce in any other plant than earlification. And there's also some work in the temperature, the development under different temperatures. So both of these biotypes, they reproduce and they continue the development in temperatures that are 64 Fahrenheit up until 21 Fahrenheit. So at the hotter temperatures, the time to develop is less, but they're still gonna develop. So we expect our beetles to be able to survive our summer and winter temperatures. When it's a little bit cooler, they do take more time to develop, but they are gonna develop. They're gonna continue. So far they're host specific and they have the ability to survive in Florida climate. They also are a very promising agent. The second one, we have Tricholobaster. This is a bat-gulling wasp, also from Australia. They create galls in seedlings and mature plants of earlification. this is our newest edition. It was imported into our labs in 2021. The same, for this one is a bit different. So Trichilogaster is not a described species yet. We're still working with taxonomists. But we know about the life cycle. It takes around 60 days. First, we start putting our adults into the plant, and then 30 days after the oviposition, we expect to see these small galls coming from the plant. And then 60 days after oviposition, we expect to see our emergence. They are very short-lived insects, so if they emerge on a Friday, we're not going to see them on Monday, but we are going to see some exit holes. And each exit holes mean that one wasp emerged. So the host range for this plant, we have done so far 47 species. We have tested native plants, we have tested endangered plants, economically important, and a bunch of other species from the same family and genus. So far, they have not created goals or surviving any other species than earleaf acacia. And this is how they look like, the goals. So it's very easy to spot on their damage. There is this study by Sanderson and collaborators. It's a brand new paper, and they studied the biomass above ground and below ground. So here the graph in the left, the picture in the left in this graph is a control where they did not expose the plants to the bad-galling wasp. And then the picture in the right are the plants that were exposed to the insect. So here we see that there is a significant difference in the growth of these plants. And then based on our studies so far, they are host specific. This wasp is well known for having an association with acacia species. There are two other sister species of Trichilogaster that are currently biological control agents in countries like South Africa, Portugal, and they are doing a really good job. So we expect that to happen with our insect as well. So far, earleaf acacia continues to expand throughout Florida, as the map that I showed that we developed the model. So we know that there are highly suitable areas for Trichilogaster where the plant is not there yet, so it continues to expand. We know there are two control methods, but also we know that those are not enough to stop spread, so we need more methods. Classical biological control is a very valuable tool that we can use, and we can combine with these other management techniques. And this research in general is providing us a really good foundation for biological control of earleaf acacia in Florida. And the only goal that we have is to provide a safe and effective biological control agent that can be used in the field, and it can help us to preserve our Florida ecosystems. So I would like to thank all the members from the Mintier with Biological Control Lab, our funding sources. And if you have any question, please send me an email or let me know. And then again, here's the QR code. If you do have time in the field, please record this plan that we're looking for the distribution still. Thank you.
Thank you very much, Sarah. Did I hear you correctly when you said for at least the first one of the biocontrol species you're looking to submit for release next summer?
Yes. Dr. Mintier is working on that. They already got in the documents, but we still need to finish with some host testing.
Are there things like letters of support that the community can provide for that? Will there be a public comment period later, or is that something that's opening soon?
That might be later. I believe that they still have to approve that, and then I'm not sure how it works, if they have like a public hearing but I just know that they have to file the petition first with all the evidence that it is a safe biological control agent.
Awesome. Thank you very much. It's exciting that we have a species, potential biocontrols for something that's not already devastating. And hopefully that'll stop.
Yeah, so that was the aim, to work in something now before it's everywhere and really out of hand. So I think we have a good shot on releasing agents that can slow the spread.
Awesome. Thank you very much. We'll move on to our final researcher for the day, Maya. You're up, ma'am.
Hi, thanks so much for inviting me. I'll do share screen. Please let me know my slides.
I think it's just taking a second to come through. Yep, I can see it now. I can see your presentation, but I can't hear you.
You're a bit cut off. Do you see my slides?
Yes, I can see your slides. I might be getting poor reception, one of us. I can see and hear.
Okay. You hear me?
Yes, you're breaking up a little bit.
Okay, I'll start, and if there's any problem, just do this, for instance. Okay. Okay, thank you. So again, thanks so much for inviting me. My name is Saar. I'm currently a postdoc at Florida Museum of Natural History. And today I want to tell you about one of my postdoc research projects, the little fire ant, low-key but destructive invasive species. And I will tell you a bit about what we think is going on in Florida specifically. And these are my dear colleagues. This research was led by Andrea Lucky from the entomology and nematology department in UF. So these are in the bottom of the slide, some ways to communicate me if you'd like, including my email and website. And this will be our roadmap for today. I added these arrows in the bottom of my slides to help you follow my talk, see what stage are we on. So I'm going to start with ants, just to give you some idea to, so we will be all of us on the same baseline. Ants are one of the most successful groups of animals in the world. They occupy most terrestrial environments, maybe except for Antarctica and some inhospitable islands. Think about this fact, between 15 to 20% of terrestrial biomass in the tropics are ants. Think about all the biomass in the tropics, about 1/5 of it are ants. They have key eco roles because they excavate their nest, they're turners of soil, they're spreader of organic matter. Because of their relationship with plants, they spread seeds and other plant material, and they impact profoundly on other organisms and ecosystems because they have eco roles, anything you can think of. They are scavengers, they are predators, such as these army ants, which you see in the picture. They are herbivores, such as leafcutter ants. They have other tight relationship with plants. And I'm showing you this to sort of give you a taste of the diversity of ants and their tools and their behaviors that help them be so successful and adapt to their natural environments. For instance, this soldier, its head is shaped like a plug to block a nest entrance. And I want you to think about how successful ants are in their native environment, and think about what happens if some of them are able to establish in invasive ranges, how successful they can be with their tools and specialized behaviors. And they do that, they are successful, and it's often at the expense of other local wildlife, and as they break the balance of local ecosystems. So I want to ask you a question. Which ant recently stung you in Florida? And I will answer because we don't have a lot of time, but I bet that recently, or if not recently, that at some point a fire ant stung you in Florida. And this is just to make a distinction. Fire ants are not little fire ants. As you can see, this is a paper from 2015, and this is a molecular analysis. And fire ants, Solenopsis sp, are not related to little fire ants. They're very far away on the tree, and their name is similar. That is because their sting reminds of a burning sensation. And you can see other differences, like size. Little fire ants are much smaller. a different color. Fire ants are often called red imported fire ants. I will just simply call it fire ant, and I'll give you the distinction: fire ants and little fire ants. So I'm working on little fire ants. They are less studied, by the way. And so they are native to South and Central America. The earliest records we have in Florida are from 1924. This is the little fire ant global distribution. In gray, you see the native range. In black, invasive range. And in X are invasive ranges in which they are greenhouse pests. So they like to be in plant nurseries, greenhouses. So the X's mean that they're not in the wild, but they exist in greenhouses. This is also to say that today the distribution of little fire ants is actually much wider than that of fire ants. And that is although they are much less studied. Fire ant is one of the most studied ants in the world, but little fire ants have a much wider distribution today. So they have invaded the US, first in Florida and after that in Hawaii. And why should we care? First of all, just like I named this presentation, little fire ants are low-key but destructive. And why I say low-key, because usually humans detect their invasion when it's already too late and they are abundant in numbers. Before they're established in strong numbers, they're small, 2 millimeters at the most. They're kind of sneaky. If you look at the field on them, they're slow. They hide. You don't notice them until it's too late, until they're already established, and it's harder to eradicate them when they're already established. And their damage is wide. They displace local arthropods in their invasion location. They disrupt plant-insect interactions, including pollination. They have a painful and an allergenic sting that may cause eye disease in humans and domestic animals. They are known to limit agricultural workforce. So in orchards where they're established, they sting workers, limit their work, and obviously cost a lot of money to their employers. In Hawaii, for instance, they estimated 20,000 ants per square meters in their invasive areas, about $13 billion in damage and control in the next few decades, if not treated. So this is all to say that little fire ants are major environmental, agricultural, and economic pests. So they cost money to conservation efforts, they cost money to agriculture, they cost money in economics of humans. So we wanted to see what's going on in Florida in terms of the invasion of the little fire ant. This ant was not studied extensively in the US until now, although it's been here around 100 years. So the goals were to map the current distribution of this ant in Florida and also to characterize the population structure. And why is that? Because this ant and other invasive ants, in their invasive ranges, sometimes their population structure is what we call a super colony. So if you look at the blue dots on the map, that's the locations that I found that are fire ants in Florida. We have 37 locations. So imagine that all of these blue dots are one huge colony, super colony, meaning that they have the same genetic basis. They're not aggressive towards each other, so they cooperate. And together with great numbers, that's how they steal other resources of other wildlife and establish their invasion more. So we wanted to see what is the population structure of this ant. It might be one super colony, like in other invasive ranges. It might be a few subpopulations. It might be each blue dot, each location is a separate unit. We don't know. So our methods were, and I'll show you two of them, and the third one is still in analysis. So first we mapped the distribution, like I said. We found 37 locations, and then we did aggressive bioassays. So imagine two blue dots. We take ants from these locations, we put them in a small arena, and we see if they're aggressive or not. And if they're aggressive, so there's reason to believe that there are few populations in Florida. If they're non-aggressive, like I said, they must cooperate then, and maybe there's a super colony in entire Florida. Lastly, we did a molecular analysis, again, to give us a hint about the genetic basis of these ants in Florida, if they're a super colony or a few subpopulations. So this is the more alarming result that we have. We knew that little fire ants are established in South Florida, in Miami, in the Keys, and they spread along the beach on the east until Orlando, the Orlando line. And today we know that they are spread until Gainesville, at least. We have 9 new locations in this new expansion, unfortunately. Lastly, and I said this is the molecular results together with the behavioral results, and I'll again point out that this is preliminary. This is one model of what we are analyzing right now, and I'm not sure this would be exactly what would be published. But you can see, look, if you look at figure A, you'll see again the distribution, the locations of where I found little fire ants. But now these locations are colored, and let's say the green dots are subpopulation 1, the red dots are subpopulation 2, as you can see in figure B. And figure C, you can see the aggressive bioassays. So the the y-axis is the aggression level, and the x-axis is the type of groups that we checked randomly all over Florida. And you can see that the highest aggression is between subpopulations. So let's say between the green and the red subpopulation, there's high aggression. There's less aggression within subpopulations, so within localities of the green population, for instance. There is still aggression, but less. Significantly different than between subpopulations, sorry. And the lowest level of aggression is between nestlings. So if you take one locality and take 2 ants from that locality, they would not be aggressive, which is expected. But we just use that as control. So to conclude, sadly, little fire ants has spread to North Central Florida, and this invasion is established because in most cases I've found queens in those locations, meaning the reproductive units of these ants exist, meaning there is continuation of this invasion. And secondly, we identified a few subpopulations of little fire ants. They share a genetic basis. They are not aggressive to each other. So this rules out super colony hypothesis. There are probably a few subpopulations. We still don't know how much subpopulations. And I'll go back just for a second to show you that if you can see, each subpopulation is not really, mostly not aggregated. Some of the red locations are in the north, some of the red locations are in the south, right? And we think that this may suggest that there are a few introductions of little fire ants into Florida, and also that they might be transferred by humans. This had happened before in other invasive ranges of little fire ants, because, like I said before, they like greenhouse, they like plant nurseries. People that drive by buy a plant unknowingly. They go home and there's another location of little fire ants in Florida. This is it. If there's any question, I'd love to answer.
We did have one question about the vectors of expansion, and you mentioned like landscaping plants already. Are there other ways that they can move around?
So if you're asking about their own spread mode, we don't think, although we see queens with wings, we don't think that they spread by themselves so far.
Okay.
Honestly, yeah, sorry.
Go ahead.
So honestly, it's probably human transports. It's yet to confirmed in following research, but it seems that way, yeah. And because it happens in other invasive ranges, we think this is how they spread.
That makes sense. Like a way to identify, or to, if you think you have them, or to keep an eye out for them. Anything we can look for to identify them?
Oh, this is a good question. Well, I think it's that you need a binoculars to firmly identify them. I suggest that I think it's a very important question, and I would be happy to spread the information. But I would need to send you pictures. Specifically, do that. Please email me, and I'll send you pictures.
That sounds good. It seems like this is a good species to help make people more aware of.
Oh yeah.
Awesome. Well, thank you very much. We're a few minutes past time. I have a couple things I want to share real quickly just to make sure everyone's up to date on a bunch of different events. So we still have a lot, even though it's the heat of summer, our SISMAs are all still kicking butt and doing lots of great work. We have several different virtual meetings. Great to be inside during this hot weather, but there's also a lot of amazing work days that are happening out there. So really impressive with the amount of work all of our SISMAs are doing. One quick announcement, we have a new position at UF that I'll be sending information out about tomorrow. We're starting an applied citizen science program. This will be a full FTE working from home based out of somewhere in the Big Bend or Panhandle area. And again, I'll send more information out on that on the Florida Invasives listserv tomorrow. We are almost done with our 2024 schedule. We just have 2 more months, and I'm beginning to plan our 2025 SISMA call schedule. So if you have any topics, please send me some ideas. Love to hear what you want to hear about, or if you have something you'd like to share, your own projects or work, please get back to me on that. That pretty much wraps it up. I want to thank everyone for making time and for hanging out with us. We didn't go too far over. Lots of great presentations today. Hope everyone has a safe end of your summer, early fall, and we'll see you next month.